When discussing DNA replication, we're introduced to a process known as PCR. This acronym stands for polymerase chain reaction and is a technique that is used to produce multiple copies of a target strand of DNA. In science, this process is used primarily in research in the medical and biological fields to multiply a target strand. Outside of science, we can see this used in fields such as agriculture to study formerly used hybrids and their genomes, and we can also see this used in areas such as forensics to increase the abundance of DNA collected from a crime scene. But how does this replication of DNA take place? Unlike natural replication, which takes place in a cell when a signal is sent to that cell, PCR takes place in a test tube within a lab when a scientist needs a larger quantity of the DNA. When completing PCR, we use an instrument known as a thermal cycler, or commonly known as a PCR machine. To use the thermal cycler, we must produce a solution in a test tube for the machine to process. So in our test tube, we're going to add our target strand of DNA, primers, free nucleotides, and DNA polymerase, along with some other buffers and additives that aren't necessarily important to understanding how the process works. So once we've prepared our test tube, we're going to place it in the thermocycler and start the machine. And at this point in the process, our job is pretty well over. But what is taking place inside of that machine? So PCR takes place in 3 steps and is repeated upwards of 50 times to produce billions of copies of the DNA strand. Throughout these steps, the temperature within the unit is adjusted to provoke the processes. So here we have an illustration of the test tube when we first place it in the thermal cycler. As said before, in this tube we have our target strands, primers, nucleotides, and DNA polymerase. In real-life PCR, we're going to see many various lengths of DNA, but for simplicity, in this model, I used one length for all of them. So once the machine is started, the solution is heated to 95 degrees Celsius. This heating of the solution causes the DNA strands to break apart and dissociate. This step is known as denaturation. Once all of our target strands have completely separated, the solution is then cooled to 55 to 65 degrees Celsius. This cooling allows the primers to attach to the 3' end of the single-stranded DNA. They must attach to the 3' end so that our new strand is synthesized in a 5' to 3' direction. And this process of the primers attaching is called annealing. Once our primers have all attached to our target strands, the machine again heats the solution to about 72 degrees Celsius. When this desired temperature is reached, the DNA polymerase is added and works along the strand, adding nucleotides. And these nucleotides, as many of you know, are adenine, guanine, thymine, and cytosine. And they're added by the polymerase according to the sequence of the target DNA because we know that they must be complementary. And this process of adding the nucleotides and extending the strand is called extension. So once our first round of extension is complete, the process starts over and the steps are repeated as many times as necessary to produce our desired quantity of our target strand of DNA. Once all of the rounds of PCR have taken place, the product that we end up with is what we now use for the earlier mentioned purposes. And for your guys' case, in the lab, will be used to determine the suspect of your crime.